Hello everyone, my name is Robert McKee. I'm a graduate research assistant in the Department of Animal and Dairy Science at the University of Georgia, and today I'm going to be discussing Orpinomyces and the fungi of the rumen. For most of their known lives, the ruminant fungi have been victims of a case of mistaken identity. First observed over 100 years ago in rumen fluid, fungal zoospores were mistaken for flagellate protozoa, which they resemble closely. It wasn't until the 1970s when a British researcher by the name of Colin Orpin observed several ruminant microbes that exhibited fungal morphologies similar to those of a certain aquatic species of Phycomycota. These included a rhizome or vegetative rhizome that produced a reproductive sporangia-like structure that emitted flagellate zoospores. Then in 1977, Orpin confirmed his suspicions when he found chitin in the cell walls of these organisms. Anaerobic fungi have since been isolated from the hindgut and feces of several herbivores, denoting their name gut fungi, but since they are the only anaerobic fungi that are known, I prefer this title. All of the known anaerobic fungi belong to one family, Neocalamastigaceae, which is comprised of 10 known genera listed here with their date of discovery. Now, these genera are separated by morphology. This includes the monocentric or polycentric thallus, that is the production of one or many sporangiopores per rhizoid, the structure of the rhizoids being filamentous or bulbous, the shape of the sporangia and the stalk which supports the sporangia, known as the sporangiopore, and also the number of flagella on the zoospores. Anaerobic fungi also exhibit genus-dependent differences in hemocellulase and cellulase activity. There are also several novel clades which have yet to be named, and these comprise the only known obligate anaerobic fungi. Anaerobic fungi exhibit a life cycle that is similar to other fungal organisms. They begin their lives as motile zoospores that have a high affinity for fibrous substrates. This may be due to a chemotaxis produced by the initial colonization of fibrous substrates by cellulolytic bacteria. After attachment of the zoospore and encystment, the zoospore germinates and a rhizoid is developed, which penetrates into the fibrous substrate. The rhizoid will then produce a reproductive sporangia in which nuclear division occurs, producing more zoospores. Now, this whole cycle takes about 24 hours. So in a rumen where washout occurs, substrate attachment is key for fungal survival. I would now like to discuss some of the unique characteristics that make anaerobic fungi so fascinating. First, they have hydrogenosomes in lieu of mitochondria, which contain pyruvate, ferredoxin, oxyreductase, and hydrogenase that are two unique enzymes allowing hydrogenosomes to produce ATP in an anoxic environment. Secondly, their genomes exhibit the lowest percentage of cytosine and guanine of any known organism. Conversely, they have high amounts of adenine and thymine. This results in low amounts of the amino acids arginine, proline, and glycine, and high amounts of lysine, phenylalanine, and tyrosine. Next, they seem to exhibit a resting stage that allows for survival in O2-rich environments. This is crucial for transfer from animal to animal in substrates such as saliva and feces. Lastly, they show extreme affinity for substrate attachment, as mentioned in the 4 slides. This increases the retention time in the rumen and allows for more complete degradation of fibrous feedstocks. On that note, the anaerobic fungi produce a plethora of enzymes and are very, very adept at biomass degradation. Estimates range from 75 to 90% of fiber hydrolysis. This is likely due to cellulosomes as well as the physical disruption of biomass structure by the rhizoids. Now, several studies have found evidence of horizontal gene transfer from multiple bacterial lineages, adding to the mysterious nature of the anaerobic fungi. Furthermore, studies have found that lignin is degraded but not metabolized by the ruminant fungi, which would make them one of the few organisms capable of this task. Several factors affect the prevalence and diversity of fungi in the rumen. Of these, diet has perhaps been studied the most, with several studies reporting a positive relationship between fiber content and the number and diversity of fungi isolated from the rumen. Sample type and timing also have an effect. Boots and others in 2012 conducted a study which examined the effect of diet type on fungi counts and diversity in the liquid and solid phases of rumen fluid. They reported that concentrate increased the number and diversity of rumen fungi in the liquid phase, while it decreased these two indices in the solid phase. They also reported that roughage had the opposite effect. This is likely evidence of the high affinity for fibrous substrates of the rumen fungi. Time after feeding also has a profound effect, and this goes back to the 24-hour life cycle, which is relatively long when compared with the life cycles of other ruminant microorganisms. Also, what you are sampling for, whether it be zoospores or rhizobia, can have an effect on quantification and diversity of rumen fungi. Lastly, species effects show large variability both in domesticated and wild ruminants and herbivores. Would now like to discuss the Orpinomyces genus of the anaerobic fungi. This genus was named for Colin Orpin, and they are described as exhibiting a polycentric thalli, filamentous rhizoids, and polyflagellated zoospores. This genus consists of 3 species as of 2015. That is O. bovis, O. intercalaris, and O. joyonii. Several studies have tried to quantify their abundance in the rumen, with Sirohi and others reporting 48% Orpinomyces of all the rumen fungi in 2013, while Kettleman and others in 2013 reported only 12%. This is likely due to varying diets and perhaps even location and herd differences, even animal-to-animal differences, much like the other ruminant microorganisms. Orpinomyces exhibits a very strange genetic makeup, and evidence has been found of horizontal gene transfer from multiple bacterial lineages. In a genome sequencing project, Youssef and others in 2013 identified several enzyme-coding genes in the genome of Orpinomyces. These included 357 glycoside hydrolases, 24 polysaccharide liases, and 92 carbohydrate esterases. The genome of Orpinomyces also contains genes for cellulosome structures, including a scaffoldin gene or scaffoldin protein. Orpinomyces have exhibited the ability to survive long exposures to oxygen in a liquid medium. think the estimate was 168 hours, which again is crucial for the transfer from animal to animal in things such as saliva and feces. In another study, Day and others in 2004 reported increased animal performance in calves fed an Orpinomyces culture compared to those that were not fed, or a control group. They also reported a lower pH, which they attributed to increased volatile fatty acid production, as well as a lower feed to gain ratio. Is my work cited? And thank you very much.